Hello, Civitans from around the country and around the globe. This is Dr. Craig Powell, Director of your Civitan International Research Center for Neurodevelopmental Disorders, bringing you yet another update on what I hope is your favorite topic, progress in our fight together to optimize the lives of children and their families challenged by neurodevelopmental disorders. It's really been a banner year for your research center. We successfully just completed a multi-center drug treatment trial for Pitt-Hopkins syndrome, a neurodevelopmental disorder caused by rare mutations in the TCF4 gene. This new drug that targets insulin-like growth factor receptors was not only proven safe for use in this population, but also highly effective in improving symptoms for these patients on multiple outcome measures, some of which were created by our own Dr. Cassandra Newsom for this study. This provides hope not only for this particular genetic disorder, but also for multiple related genetic forms of neurodevelopmental disabilities. This same drug is being tested in many other types of neurodevelopmental disorders at UAB and around the country. This year, we reimagined the McNulty Civitan Scientist Award, funded by the Foundation for Children with Intellectual and Developmental Disabilities, or FCIDD. Our goal was to promote multidisciplinary collaborations among Civitan scientists from disparate areas of research in the hopes of providing preliminary data for much larger future grant applications with multiple scientists working together to solve important challenges. We received no less than 19 separate proposals written by more than 40 different scientists working together across disciplines as diverse as artificial intelligence, virtual mindfulness apps, and rodent disease models research. Clearly, the appetite for teaming up across research areas is stronger than ever here at your Civitan International Research Center. As always, we hope this video will inform, entertain, and inspire you all to double down on your efforts in support of your Civitan International Research Center and the work that we do every day on behalf of children and their families.
Our lab is unique at UAB in that we don't have just one line of research. We support research in multiple areas of neurodevelopment. We are part of a 25-site consortium across the country enrolling 7,500 women in their second trimester of pregnancy. We're following them and their babies until they're 10 years of age, trying to better understand how the infant brain develops, what kinds of things support healthy brain development and resiliency, and what kind of events might cause changes in the developmental trajectory. Our project enrolls children who have a neurodevelopmental disorder, and we do extensive behavioral testing with those children to better understand their strengths and challenges. We provide a diagnosis to the families when that's appropriate, and they can use that diagnosis to access resources. As part of the Healthy Brain Child Development Study, we're using several cutting-edge techniques to understand infant brain development, including MRIs as early as 1 month of age, EEGs that help us measure brain electrical activity in response to different stimuli in babies, eye tracking devices that help us understand how they're learning numbers and words and gaining executive functioning skills. So our research serves two purposes. One is to provide a clinical service to families in the community. So we provide diagnostic evaluations for children with autism. But our goal is also focused on developing new treatments and assessing new treatments for rare genetic conditions and autism.
So the dentate gyrus is one of the few regions in the mammalian brain where there are stem cells that continuously produce new neurons. And just by chance, we came across a new technique to identify these adult-born cells, since there's not very many of them. And so this made me interested in the process of adult neurogenesis, and we've been studying these adult-born cells ever since. So we think that understanding how neurogenesis works in the normal brain will provide new information to understand how this process goes wrong and what ways we can counteract it when these processes go wrong in many different diseases. So we try to think of individual neurons as information processing units. And so we're interested in how they receive information from other neurons, so how their synapses work. We are interested in how they process that information. So we ask questions about the intrinsic excitable properties of individual neurons. And we're also interested in how individual neurons communicate with the surrounding neurons. And so we're interested in their synaptic output. And so we try to take this sort of comprehensive approach to think about how individual neurons contribute to the properties of the entire circuit of the dentate gyrus. And so one of the main goals of my research program is to understand how these newborn neurons contribute to these various cognitive functions. So in our lab, we train students to learn about fundamental aspects of synaptic function and neural excitability. I mean, it's really providing an understanding of how neurons work, how they communicate with each other, how synapses work. And this is foundational information that really can be applied to any type of neuroscience research in the future.
So the main goals and ideas are really to investigate environmental health risk factors. These can be chemicals in the environment that we released or produced or that come out of smokestacks, heavy metals that can be found in drinking water, for example, and just sort of what impacts they have on human health, especially neurodevelopmental health, children, neurological health in adults, and I'm also interested in environmentally induced cancers. Many of the chemicals that are released into the environment, certainly many of the heavy metals that are not essential to the body, things like lead and mercury, we know have toxic effects on the neurological system. So we are specifically focusing on environmental health factors in our field. It makes us a little bit different. It's also a good collaborative area to work in terms of people that are looking at genetics or diet, because then we can kind of explore the interactions between these different components that we think are important. So we're taking advantage of some new sampling technologies that take a much smaller amount of a biological sample. So we often take blood, hair, urine, and things like that to measure exposures. And so some of these new sampling technologies should make it much easier for us to collect samples. Whether it be children's neurodevelopmental problem or adult neurological disease, is once we have a better foundation for what environmental health risk factors and to what extent these factors impact these disease processes, we can then develop policy around that to mitigate or perhaps prevent such exposures.
My lab is unique in the sense that we're not rooted in any one particular disease or brain structure, although we do study the cerebellum as a model system. I've always been interested in fundamental aspects of mechanisms and synapses, and I'm one of those kids that grew up always taking things apart. I don't necessarily could put them back together, but I really like to see how they worked. And so a lot of neurodevelopmental and neurodegenerative diseases have their basis in synaptic dysfunction. And so it stands to reason that to understand those processes, you kind of have to understand how they work in the normal brain. And that's what we do. So we don't per se study diseases, but we try to study synapses that might go awry during those disorders. There's been a renaissance in studying the cerebellum over the last few years, not only because it's involved in fine motor movement, but also because it's involved in diseases such as autism and autism spectrum disorders. Experiments in the lab use a multidisciplinary approach to doing science. We use electrophysiological and imaging tools to probe how synapses and glia work in the brain. One of the cutting-edge techniques that we use is two-photon microscopy. Two-photon microscopy allows you to have exquisite detail of individual synapses, much more so than other techniques such as confocal or wide field imaging. So a student could expect to learn all of these techniques and then go on to apply them in whatever they are interested in down the line. We try to make the lab a place where people can learn. we're always growing. I feel like I've been here for now going on 18 years, and every day I learn something, not only from my colleagues, but most importantly from the people in the lab. And I really like that about the job.